Please subscribe me and join with me. Now we are talking about the structure of DNA. The structure was founded by Watson and Crick, who was the two scientists. According to their observation, they called this structure was double helix structure. This DNA molecule, if we consider this DNA molecule, we can observe six major characteristics of this DNA molecule. First one is wound in a right hand like a clockwise. Right hand means clockwise. These antiparallel two strands one was 5 prime into 3 prime end and another one is 3 prime into 5 prime is called two antiparallel chains. If we consider this DNA molecule, we can observe four complementary base pairs like adenine and thymine and cytosine and guanine. If we consider the RNA molecules, RNA observe according to the thymine, but RNA has uracil for complementary base pairs. If we consider this complementary base pairs, adenine and thymine bond with lower energy two hydrogen bonds. Adenine and thymine bond with two hydrogen bonds. These hydrogen bonds are low energy. According to this structure, you can see it. As well as we consider the cytosine and guanine. Cytosine and guanine bond with three hydrogen bond like this. These complementary base pairs shows adenine to thymine, cytosine to guanine. According to this, thymine and adenine and guanine and cytosine. If we consider this structure of DNA molecule, one nucleotide is we can observe one nucleotide like this structure in the whiteboard. We can observe pentose sugar and phosphate group and nitrogenase base like adenine, thymine, cytosine, guanine. These two stranded antiparallel like 5 prime into 3 prime end, 3 prime into 5 prime end. If we consider structure of DNA like a part, base pairs are 1.34 nanometer per apart. Therefore, we can 10 bases include apart 3.4 nanometer for 10 bases. Sugar phosphate backbones are not equally spaced like this figure. Consider this figure. Sugar phosphate backbones called not equally spaced result in major and minor grooves. Major groove is the wider one and minor groove is the narrow one. This structure of DNA was founded by Watson and Crick. The chain running in the opposite direction, that is antiparallel. Antiparallel means 5 prime to 3 prime and 3 prime to 5 prime end like this. Two base pair, that is the which white purine and pyrimidine. Purine is called adenine and guanine. Pyrimidine is thymine and cytosine. Purine is adenine and thymine. Pyrimidine is cytosine and thymine. These two purines would be too large and two pyrimidines too small. The span of gap between the two chains along the axis of the molecule base pair are 1.234 nanometer for apart according for the regulatory indicated X-ray directions. The complete turn of the double helix compares 3.4 nanometer for 10 bases. Like that, two chains are said to be complementary because the sequence of Watson and Crick published their model in the Maurice Wilkins were awarded by the Nobel Prize. If we consider about this DNA molecule, DNA has the major 6 characteristic of like this wound in right hand and base pair 0.34 nanometer apart, sugar phosphate backbone are not equally spaced, therefore resulting major and minor groups in this DNA molecule. Sugar phosphate backbone you can see linked with phosphodiester bond with sugar phosphate backbone. We can see like this phosphate and pentose sugar was linked with phosphodiester bond. These hydrogen bonds are lower energy hydrogens because the strand was long distance. Therefore, hydrogen bonds get lower energy for these bonds. But if we consider the phosphodiester bonds, these are much energy hydrogen bonds like this. Meanwhile, James Watson and Francis Crick of the laboratory in Cambridge had chosen what was the successful approach. Using this all chemical and physical information they could gather, they began building scale model of polynucleotide in the hope that convincing structure would emerge. Watson's book Double Helix provides fascinating insight into their works. Two lines evidence provide real. Firstly, they were in regular communication with Wilkins and had access to X-ray diffraction data against which they were able to their test models. These data strongly suggested helical structure with regular spacing 0.34 nanometer along its axis. Secondly, they released the significance of some evidence published. Irving Chargaff's rules means called 10 bases include 3.4 nanometer. Chargaff's rules is much helpful for observe double helix structure with Watson and Crick. Today we are talking about structure of DNA. Next we talking about the DNA replication. DNA replication is much interesting side. They has some enzymes called helicase, ligase, polymerase, and like this. If we consider the structure of DNA, what are the major characteristic of DNA molecule, and then we talk about DNA replication. Okay, please like this video.